Hello, and welcome. Today, we're going to dive into the fascinating world of the Human Genome Project. The Human Genome Project was an international research effort that aimed to identify and map all the genes in the human body. It was a monumental undertaking, spanning over a decade and involving thousands of scientists from around the world. To understand the significance of this project, let's first discuss what the genome is. The genome refers to the complete set of genetic material, or DNA, in an organism. It contains all the information needed for the development and functioning of that organism. In the case of humans, our genome consists of about 3 billion base pairs of DNA. The Human Genome Project was a groundbreaking scientific effort to map the entire human genome. It began in 1990 and was completed in 2003, involving scientists from around the world working collaboratively. The goal was to identify all the genes that make up human DNA, an immensely complex and ambitious undertaking. This project has revolutionized our understanding of the human genetic code and paved the way for advancements in medicine, biology, and biotechnology. The image on the slide depicts a close-up view of a human chromosome with a beautiful, intricate visual emphasizing the double helix structure. The human genome is the complete set of genetic information that makes us who we are. In the 1980s, scientists began discussing the idea of mapping and sequencing the entire human genome to better understand human biology and health. The Human Genome Project officially launched in 1990 with the ambitious goal of creating a complete map of the human genome. This massive undertaking involved both public and private efforts, including the National Institutes of Health and the private company Celera Genomics. After over a decade of work, the Human Genome Project was declared complete in 2003, marking a major milestone in our understanding of human genetics. This groundbreaking achievement has paved the way for countless advancements in medicine, biology, and our overall knowledge of the human body. The Human Genome Project was a landmark scientific endeavor aimed at mapping the complete set of human genes, providing a detailed blueprint of our genetic makeup. A key objective was to identify genes associated with various diseases, which could pave the way for new treatments and prevention strategies. The project also spurred the development of advanced sequencing and bioinformatic technologies, significantly advancing genetic research and its real-world applications. By analyzing the human genome, researchers hoped to gain valuable insights into human evolution, our genetic history, and our relationships with other species. These goals and objectives highlight the transformative potential of the Human Genome Project in expanding our understanding of human biology and health. The human genome is an incredibly complex and intricate system made up of billions of base pairs of DNA. Over the past few decades, scientists have made incredible progress in mapping and understanding this genetic code. This slide outlines some of the key milestones in the journey to map the human genome. First, researchers identified unique DNA sequences called genetic markers, like short tandem repeats, that could act as signposts along the genome. By tracking how these markers were inherited in families, scientists were able to create a genetic map showing the relative positions of different genes. The next step was physical mapping, which involved directly locating and ordering genes on the chromosomes using techniques like cloning and chromosome banding. This allowed scientists to build a more detailed, comprehensive picture of the human genome and how the different genes are organized and connected. The human genome is made up of DNA, which we need to break down into smaller fragments for analysis. We then insert these DNA fragments into vectors, which allow us to replicate them. Next, we determine the order of nucleotides in each fragment through sequencing. Finally, we align and assemble all the sequence fragments to reconstruct the entire human genome. The Human Genome Project used a variety of sequencing technologies, including Sanger sequencing and next-generation sequencing, to generate a massive amount of data on the human genome. This data has provided valuable insights into the organization, function, and evolution of the human genome. The Human Genome Project relied on an older, time-consuming sequencing method called Sanger sequencing. But technological advancements have revolutionized genome sequencing, making it faster, more efficient, and more cost-effective. The next-generation sequencing, NGS, Platforms like Illumina have enabled large-scale genome sequencing projects, including population studies and personalized medicine. Third-generation sequencing technologies like PacBio and Oxford Nanopore can generate longer DNA reads, allowing for the sequencing of complex genomic regions. These technological breakthroughs have transformed the field of genome sequencing, paving the way for groundbreaking discoveries and applications. Bioinformatics is the field that applies computational methods to analyze and manage large genomic datasets. Key areas include data storage and management, sequence alignment, gene prediction, and statistical analysis/visualization. Massive datasets from DNA sequencing require specialized systems to handle the volume and complexity. Sequence alignment tools compare DNA sequences to identify similarities, differences, and evolutionary relationships. Gene prediction algorithms locate the position and function of genes within genomes. Statistical analysis and visualization techniques are used to uncover patterns and trends in genomic data. These bioinformatics approaches help researchers understand genetic variation and its implications for health and disease. Genomic data is revolutionizing various fields, from personalized medicine to forensic science. In personalized medicine, we can now tailor treatments based on an individual's genetic profile, improving efficacy and reducing side effects. A genome sequencing also allows us to identify genetic predispositions to diseases, enabling early detection, preventative measures, and targeted therapies. DNA profiling using genomic data plays a crucial role in criminal investigations, helping to identify suspects and exonerate the wrongly accused. In agriculture, genomic data facilitates the development of disease-resistant crops, enhancing yields and improving food security for a growing population. These are just a few examples of the transformative applications of genomic data that are shaping our world. Personalized medicine is about tailoring treatments to individual patients based on their unique genetic, environmental, and lifestyle factors. The goal is to optimize treatment outcomes and avoid adverse reactions. Pharmacogenomics specifically looks at how genetic variations impact how people respond to different drugs. By understanding these genetic differences, healthcare providers can better predict drug effectiveness and potential side effects for each patient. This allows for more personalized and effective treatment plans. Genetic disorders are caused by mutations in specific genes that affect protein function and lead to disease. Genetic variations can increase an individual's risk of developing certain diseases like cancer or heart disease. Genetic testing can help diagnose genetic disorders, assess disease risk, and identify potential treatment options. Emerging gene therapies aim to correct genetic defects, offering hope for treating and even curing genetic disorders. These topics will provide an overview of how genetic factors contribute to disease and how genetic testing and therapies are advancing. Protecting individuals' genetic information is crucial to prevent discrimination based on their genes. Participants in genomic research must fully understand the risks and benefits and have the right to refuse participation. Policies are needed to prevent insurers or employers from using genetic information to discriminate against individuals. Genomic research raises complex ethical questions around prenatal testing, genetic screening, and reproductive decisions that we'll need to carefully consider. Genomic data is highly sensitive and personal information that requires robust security measures to protect it from unauthorized access or misuse. Secure storage and access controls are critical to ensure the confidentiality of this data and prevent it from being disclosed without the individual's consent. Informed consent is essential when individuals participate in genomic research. They must be fully aware of the potential risks and benefits of sharing their genetic information. Anonymizing genomic data by removing identifying information is crucial to protect individual privacy and prevent discrimination based on genetic characteristics. Strong legal frameworks and regulations are necessary to govern the collection, storage, and use of genomic data, ensuring it is handled responsibly and in accordance with ethical principles. The Human Genome Project was a massive scientific undertaking that required collaboration from researchers and institutions around the world. Scientists and organizations from numerous countries came together with the shared goal of mapping the human genome. This global collaboration allowed for the pooling of resources, expertise, and data, which accelerated the progress of the project. The success of the Human Genome Project demonstrated the power of international cooperation in tackling complex scientific challenges. The project highlighted the need for continued global partnerships in scientific research to drive innovation and make groundbreaking discoveries. The Human Genome Project was a massive scientific endeavor that required substantial funding and international collaboration. The majority of the funding, around 96%, came from two U.S. government agencies, the National Institutes of Health, NIH, at 58%, and the Department of Energy, DOE, at 38%. The Wellcome Trust, a UK-based charity, provided 3% of the funding, while other international organizations contributed the remaining 1%. This slide breaks down the percentage contributions from each of these key funding sources, highlighting the critical role played by the U.S. government in enabling this groundbreaking project. The Human Genome Project was a landmark scientific achievement that had a profound impact on various fields of research and discovery. It provided a comprehensive map of the human genome, which accelerated research in diverse areas, leading to a significant increase in scientific breakthroughs. The vast amount of data generated by the project opened up new research avenues, enabling scientists to study genetic variations, disease mechanisms, and evolutionary relationships in greater depth. The knowledge gained from the project also facilitated the development of new diagnostic tools and personalized medicine approaches for various diseases, improving patient outcomes. Furthermore, the project contributed to advancements in drug discovery and development, helping researchers identify potential drug targets and develop more effective therapies. The Human Genome Project has provided valuable insights into the evolutionary history of humans and our relationship to other species. By analyzing human genetic sequences and comparing them to other organisms, researchers can reconstruct phylogenetic trees and trace the origins and migrations of human populations throughout history. The project also reveals how genetic variations contribute to adaptation to different environments, shedding light on the selective pressures that have shaped human evolution over time. Understanding these evolutionary relationships, genetic ancestry, and adaptive insights is crucial for advancing our knowledge of human origins and development. Advancements in reproductive health and genetics have expanded options and empowered individuals to make informed choices about their families. Pre-implantation genetic diagnosis, PGD, allows screening of embryos for genetic disorders before implantation, enabling parents to make informed decisions. Assisted reproductive technologies, ART, like IVF and ICSI, have expanded reproductive options for those facing infertility, improving their chances of conceiving. Carrier screening identifies individuals carrying recessive genetic disorders, informing family planning and prenatal testing decisions. Genetic counseling equips people to understand their genetic risks and make informed choices about their reproductive health. Genomic data has become a powerful tool in forensic investigations. DNA analysis can be used to link individuals to crime scenes by comparing profiles. This can help identify potential suspects by matching crime scene DNA to known offenders. Genomic data can also be used to determine biological relationships, like in paternity disputes. Identifying victims is crucial, and DNA can be used to identify bodies even if they are unrecognizable. Advances in sequencing have made it possible to revisit old cold cases and obtain DNA profiles from older samples. These DNA profiles can then be compared to modern databases to potentially solve cases that have gone unsolved for years. Genetic variation within and between populations can reveal patterns of migration, gene flow, and isolation. Understanding population structure is crucial for studying evolutionary processes like natural selection, genetic drift, and mutation. Genetic diversity is essential for the survival and adaptation of species, especially as they face environmental changes. Genetic markers can help trace ancestral lineages and provide insights into human migrations and population history. Epigenetics explores how environmental factors can alter gene expression without changing the underlying DNA sequence. These environmental factors include diet, stress, and exposure to toxins. Epigenetic modifications involve chemical tags that attach to DNA or associated proteins, affecting gene activity. These modifications can be inherited or influenced by external factors. Epigenetic changes have profound implications for health, contributing to chronic diseases, cancer, and developmental disorders. Understanding these epigenetic changes is crucial for personalized medicine and disease prevention. The human microbiome is a vast and complex ecosystem consisting of trillions of microbes, mainly bacteria, that reside in our gut, skin, and other parts of the body. These microbes play a crucial role in maintaining a healthy immune system, aiding digestion, and contributing to our overall well-being. Emerging research is shedding light on the intricate interactions between the microbiome and various aspects of human health, including conditions like obesity, inflammatory bowel disease, and even mental health. Understanding the microbiome and its impact on our health is an exciting and rapidly evolving field of study that holds great promise for improving our overall well-being. Nutrigenomics is the study of how our genes influence the way our bodies respond to different foods and nutrients. By analyzing an individual's unique genetic profile, we can create personalized nutrition plans to optimize their health and well-being. Nutrigenomics can help identify genetic predispositions to certain chronic diseases, allowing us to guide dietary choices to mitigate those risks. Genetic testing can also reveal individual differences in the ability to absorb specific nutrients, enabling us to tailor dietary interventions accordingly. Personalized nutrition powered by nutrigenomics holds great promise for empowering individuals to take control of their health through customized dietary approaches. Stem cells have incredible potential to regenerate damaged tissues and organs. This offers hope for treating devastating conditions like Parkinson's and spinal cord injuries. Stem cell research is revolutionizing medicine, leading to new therapies and treatments. Clinical trials are underway to evaluate the safety and effectiveness of stem cell therapies. These trials are an important step towards the widespread adoption of these transformative treatments. CRISPR-Cas9 is a groundbreaking gene editing tool that allows us to precisely target and modify DNA sequences with unprecedented accuracy. This technology has revolutionized the field of genetics, enabling scientists to make targeted changes to genes. CRISPR-Cas9 holds immense potential for treating genetic diseases by correcting faulty genes or introducing new ones. We've seen promising results in using CRISPR to treat inherited disorders like cystic fibrosis and sickle cell anemia. The ability to precisely edit genes opens up new avenues for developing personalized, targeted therapies for a wide range of genetic conditions. The Human Genome Project has paved the way for groundbreaking advancements in various fields, including medicine, agriculture, and forensics. As technology continues to evolve, we can expect even more transformative discoveries in the years to come. Some of the key future directions and emerging trends include personalized medicine, tailored treatments based on an individual's genetic makeup, gene editing, modifying genes to treat diseases and enhance traits, synthetic biology, engineering new organisms with specific functions, next-generation sequencing, faster and more affordable genome sequencing. These advancements hold immense potential for addressing global challenges such as disease prevention, food security, and environmental sustainability. The implications of genetic information on both personal and societal levels need to be carefully considered and debated as we navigate the ethical landscape of genomics. Public understanding of genetics is essential for informed decision-making and fostering responsible applications of genomic technologies. Effective education and outreach efforts are crucial. Ensuring equitable access to genomic technologies and their benefits is crucial to address disparities and promote social justice. We must work to create a more inclusive and equitable future. Clear policies and regulations are necessary to guide the responsible use of genomic data and protect individual privacy. This will be an ongoing process as the field continues to evolve. The Human Genome Project has been a groundbreaking scientific endeavor, unlocking the secrets of our genetic blueprint and revolutionizing our understanding of human biology. This wealth of genomic data offers unprecedented insights, impacting fields like medicine, healthcare, and scientific research in profound ways. As we harness the power of genomics, we must also consider the ethical implications and ensure responsible applications that benefit society as a whole. Looking ahead, the future of genomics holds immense promise, but also requires careful navigation to ensure equitable access and positive societal outcomes.